The third type of RNA is known as the transfer RNA. Transfer RNA, abbreviated as tRNA. tRNA. Now, tRNAs are the smallest of RNA molecules. They have about 70 to 90 nucleotides. Although the structure of tRNA is complicated, we represent it like this. We represent it like this. Okay, so this is the 3' end and this is the 5' end. At the 3' end, remember this is the OH of the 3', right? At the 3' end, there is a sequence of 3 bases, A, C, C. Okay, this is known as the acceptor stem. This is known as the sequence of 3 bases is known as the acceptor stem. Okay, what is the purpose of the acceptor stem? An enzyme will attach the amino acid by forming an ester bond with this. Okay, now I'll get to that in just a minute. So, tRNA, the function of tRNA is to bring amino acids to the ribosomes for protein synthesis. Okay, the function of tRNA, okay, is to bring amino acids to the ribosome for protein synthesis. What does that mean? What it does is it interprets the genetic information from mRNA. Remember, mRNA has the genetic information, right? mRNA has the genetic information, right? Essentially, what is this genetic information? Genetic information is nothing but the sequence of amino acids in proteins, right? So, but then mRNA is a combination of nucleotides, right? One nucleotide and then the other one and then the other one. So, this needs to be converted to amino acid sequence, right? So, this genetic information is available in mRNA as a sequence of nucleotides. Only tRNA can translate the genetic information. tRNA can translate this genetic information into a sequence of amino acid that can be made into proteins. So, tRNA translates the genetic information into amino acids. So, going back to the structure of tRNA, so this is what the structure, we drew it before, a loop here and a loop here and a loop here, right? So now why are these loops being formed? Because there are in some areas hydrogen bond between complementary base pairs is being formed, so there are loops in tRNA. So this OH, where the acceptor stem is, the sequence ACC. Remember, we talked about protein synthesis in the last slide. So the amino acid according to the sequence that the tRNA has interpreted. An enzyme attaches the amino acid by forming an ester bond with this. Okay, and there is also, in every tRNA, there is an anticodon here. Okay, there is a anticodon in this region. Anticodon. And what's an anticodon? Anticodon is a series of 3 bases. It's a series of 3 bases, and these bases complement 3 bases on the mRNA. So it's a 3 base sequence, and these bases complement the bases on mRNA.